It's talking. So welcome everybody. We have today a back-to-back exit seminars from Alok and Monash. Alok is first up. Alok Pradhan joined the Centre for Cryo-Electron Microscopy for Membrane Proteins doctoral training program in April 2022, following completion of undergraduate studies at University of Wollongong. So as an industrial transformation training centre student, Alok has been involved in a project that has an industry partner, and that industry partner is Dimerix. Dimerix is a clinical stage local biotechnology company, currently focused on renal and pulmonary disease, and the use of co-receptor targeting in particular to provide novel treatments. So as part of the doctoral training program, Alok also has had the opportunity to do an internship with Dimerix, and his internship was involved with the business development side, so things like pipeline analysis, competitive landscape analysis, target product profiles, etc. And because Dimerix is a relatively small company, but yet with an advanced pipeline, that has given the opportunity to work both with preclinical people, but also their clinical development people and their regulatory group. So giving broad experience in different aspects of that process. So today, though, Alok's going to present on the research work that he's done as part of his thesis, where he's worked on 3 clinically important receptors: the angiotensin 1 receptor, the CCR2 chemokine receptor, and the CXCR2 chemokine receptor, where he has explored pharmacological and biochemical aspects of G-protein interactions. With that, I'm going to hand over to Alok, and we look forward to hearing your.
Yeah, thank you for the introduction, Patrick. I'll get started. Let me get my pointer up. Yep. So the topic of my presentation is pharmacological profiling of G-protein promiscuity in AT1R, CCR2, and CXCR2, and biochemical optimization of AT1R, CCR2 for structural studies. So first, I'd like to begin by introducing GPCRs, or G-protein coupled receptors. These are large family of cell surface receptors that detect external signals and transmit them into cell to produce responses. GPCRs are highly important both biologically and clinically, with around 1/3 of the FDA-approved drugs targeting them. As shown in the figure, they are activated by a broad range of signals.
Are you sharing the slides on Zoom?
Yeah.
Sorry.
Sharing through Zoom.
Sorry. Give me a second.
Yeah, that should be good. Yeah.
Oh. Is it more?
Okay.
Yes. Good job. Excellent.
Where was I? Yes, as shown in the figure, they are activated by a very broad range of, sorry, where's my pointer now? Yep. Sorry. As shown in the figure, they are activated by a broad range of signals, including ions, bioenergetic amines, lipids, peptides, proteins, and even sensory stimuli such as light, odorants. Because of this, GPCRs are involved in different physiological processes. Once activated, they initiate a range of intracellular signaling pathways that lead to different cellular effects. GPCR signaling pathway is mainly mediated by heteromeric G proteins, which are made up of the alpha subunit, the beta, and the gamma subunit. And depending on the alpha subunit, different downstream effectors and secondary messengers are activated, leading to cellular responses. And although GPCR signaling is mainly mediated by G proteins, GPCR can also signal through other transducers. I talked about GPCR, but I also wanted to talk a bit about the G protein cycle, as it will make it easier for us to understand the experiments that I'll be explaining in a while. So the G protein cycles starts at the cell membrane where we have an inactive receptor and a G protein heterotrimer. When the ligand activates the receptor, the G protein is recruited by the receptor, and the G protein at this stage has a GDP bound to it. The activation of the receptor causes the G protein to release the GDP and attach a GTP molecule. And once the GTP is attached to the G alpha, the G alpha subunit and the G beta gamma subunit dissociate and activate different effectors to carry out cellular responses. The GTP in this alpha subunit hydrolyzes to GDP, and it goes back to forming a heterotrimer in the membrane, and that is how the cycle keeps going. The stage of the G protein cycle that we are interested in at this stage is the stage 3, where the bound GDP has been released, but the GTP has not yet been bound. It is also called the G0 state, and it gives us a state which is highly stable for structural as well as pharmacological studies. So I will first introduce some of the GPCRs that I'm working with for my project. The class A angiotensin receptor type 1 is expressed widely throughout the body and is present in all organs, including the heart, kidney, liver, adrenal, brain, and lungs. It mediates most of its action of its agonist, the angiotensin 2, and it primarily signals via GQ. It is present in all organs of the body, and including the blood vessels, the kidney, heart, adrenal glands, and brain, and carries out various different functions in all these different organs. The chemokine receptor are a class of class A GPCRs, which play an important part in our immune system, and they are responsible for chemotaxis of immune cells to the site of infection and inflammation. The CCR2 or the CC motif chemokine receptor 2 plays a pivotal role in the immune system and mediates recruitment of monocyte and macrophages to inflammatory sites. It is the main receptor for its ligand, the CCL2, which is also called MCP1, and it primarily couples to the G alpha I family of G proteins. It is involved in many disease indications of the heart, kidney, lungs, liver, etc. And some of the examples of the disease that's involved in is cardiac fibrosis, glomerulonephritis, and rheumatoid arthritis. The other receptor that I'm working with is the CXC motif chemokine receptor 2 or CXCR2. CXCR2 mediates most of the chemotaxis of leukocytes and also regulates wound healing, angiogenesis, and inflammation. It is the main receptor for CXCL8, or it's also known as interleukin 8 or IL-8, and it also couples to primarily GI and is involved in many disease indications of the heart, kidney, lungs, liver, etc. And some of the disease indications include asthma, cystic fibrosis, and different types of cancer. So one of the main reasons that I'm focused on these three receptors, the AT1R, the CCR2, and the CXCR2, is that besides being an important drug target, they are also directly relevant to my industry partner, Dimerix. Dimerix is developing the DMX-200 for FSGS, which is a CCR2 antagonist used together with the AT1R blocker and is currently in phase 3 trials. One of the hypotheses behind this project is that these receptors and potentially others may function together, potentially forming heteromers. And with that in mind, the first step was to establish a broader signaling profile of the target receptors. And once those profiles were defined, one of the initial aims of the project was to test for potential functional heteromers. And if found for the evidence, then that would support for further structural studies of the heteromeric complexes. So based on that rationale, the project had three main aims. The first aim was to assess the pharmacology of individual receptors, specifically their G protein recruitment profiles, as this could guide us the later structural biology work. And within this aim, I first identified the optimal assay system and format for probing G protein recruitment to AT1R, CCR2, and CXCR2. And I finally used that on a heteromer-based assay to test whether functional heteromer could be observed between AT1R, CCR2, and CXCR2. The next part of the project was then informed by pharmacology results. As you'll see later, because we did not obtain clear evidence for the functional heteromerization of these receptors, the structural work was ultimately focused on generating novel complexes. of the individual receptors, specifically the AT1R with GQ with angiotensin 2 and the CCR2 with GI with CCL7. Which brings me to my first aim. Before I begin, I first cloned these constructs for my structural biology as well as pharmacological studies. All these receptors have a similar architecture with a N-terminal HA signal peptide, a flag tag, which is used for both detection as well as purification, and the receptor flanked by 3C cleavage sites on both ends, and a C-terminal tag. So the 8-his is like 8 histidine tags is used for the purification of monomer structures. The large bit and high bit is going to be used to provide additional stability during complex purification if required. The NLUC and venous construct will be used for the pharmacology of receptor monomers as well as heteromers. And similarly, the 8-his and large bit and high bit clones were cloned in both pFastback as well as pcDNA for structural as well as pharmacological studies, whereas the NLUC and venous constructs are only cloned in pcDNA they're only being used for pharmacological studies. So once I cloned all those constructs, I used the TruePath G protein dissociation assay to validate the 8-his large bit and high bit construct. So in this assay, the G alpha subunit is tagged with the RLEC8 and the G gamma subunit is tagged with the GFP. In the inactive state, the G protein remains as a heterotrimer. So the donor and acceptor are in close proximity, and that gives a high Brett signal. When an agonist activates the receptor, the G protein is activated, and the relative arrangement of the subunit changes, leading to a decrease in the Brett. So this assay allows us to monitor the receptor-mediated G protein activation in real time. And based on what is already known in the literature, AT1R preferentially couples to GQ proteins, and CCR2 and CXCR2 preferentially couple to the GIO family of proteins. And this was the finding of the TruePath assay. So the AT1R and CXCR2 constructs behave like wild type, very similarly to wild type, whereas the CXCR2 high bit and to some lesser extent the large bit have reduced windows. However, the potencies of these constructs remain similar. So before I could profile G protein recruitment across the receptor, I first needed to establish an assay system that could measure recruitment to target receptors. And so in this part of the project, I optimized 3 aspects of the assay using GQ for AT1R and GI2 for CCR2 and CXCR2, as they are the canonical couplers for those receptors. And all these experiments were carried out in all these Brett experiments were carried out in delta GOL HEC293 cells, where all the endogenous G alpha subunits have been knocked out. So the first thing I optimized was the assay format. I compared whole cell condition with permeabilized cell treated with a pyrase. So in whole cells, intracellular guanine nucleotides are present as normal, so the G protein cycle can continue as normal. However, in permeabilized cells treated with apyrase, the guanine nucleotides are depleted. So it should lead to a more stable G protein complex. The second thing that I optimized was the type of G protein used. Here, I compared wild type and dominant negative G proteins. Wild type G proteins undergo normal GDP-GTP exchange, where dominant negative G proteins contain targeted mutations that reduce their affinity for guanine nucleotide and impair their exchange. And the third thing that I optimized was tag placement on the G protein. I compared tagging the G alpha subunit with tagging the G beta gamma to determine which format gave me the best signal window and the most reliable data. So here I'm showing you one of the examples of the assay optimized using the CXCR2 with GI. In this experiment, the CXCR2 was tagged with an NLUC and the beta gamma subunit was tagged with a venous, and we were using wild type versus dominant negative G protein. On the left panel, we have the wild type GI, and on the right, we have the dominant negative GI. On the top panel, I'm comparing whole cells, and on the middle, I'm comparing permeabilized cells, and then at the bottom, I'm comparing the permeabilized cells added with apirase. From this data, we can see that the wild-type GI has a downward response as it has a faster nucleotide turnover, and the dominant negative G protein has a positive signal due to the mutation that don't allow it for the G protein turnover. And I also plotted the CRCs, and also we didn't observe much difference between the whole cells compared to the permeabilized cell with apirase. So in the previous example, the dominant negative G protein appeared promising because it gave us a positive signal and a good signal window. However, when I extended this comparison to other dominant negative G protein subtypes, I found that that was not consistent across all of them. For example, this example dataset, I tested AT1R with 5 different dominant negative G proteins, and while GI2 did give me a positive response, the GS response was biphasic, and others do not have an interpretable response. Similarly, on the right, I tested the G alpha tagged with NLOC. These two are the examples that I'm using here. I did it for the entire panel, but it did not produce any robust signal as compared to using the beta gamma tagged Venus. So based on these optimization experiments, I selected whole cell condition, wild type G proteins, and beta gamma tagged format for the recruitment assay. And this combination gave me the most robust and reproducible data overall, and therefore was used in subsequent profiling studies. So once I picked the assay format and system, I needed to test out the recruitment to the receptor. And for that, I picked out the G protein based on where the different receptors and G proteins are expressed. So AT1R is predominantly expressed in most tissues. CCR2 is mainly found in monocyte macrophages and activated T cells. CXCR2 is expressed mainly in neutrophils. And all these 3 receptors are also expressed in the neurons in the central nervous system. So once I found the location of the places where the receptors are expressed, I looked at all the different G protein where they're expressed, and based on that, I picked 12 different G proteins from all the different families of G proteins. And now we have a panel of 12 different G proteins, and I have 3 different receptors. So the way I'm going to be running my assay is that I have a receptor tagged with NLOC, wild type G alpha, and a beta gamma tagged with Venus. When I add in a ligand, it activates the receptor, which causes, it activates the receptor, and then upon that, it increases the proximity of the G protein with the receptor. And in the presence of fumarate, we get a Brett signal that can be recorded. And so in the recruitment assay, I observed mainly 3 kinds of responses with the receptor and the different G proteins that I tested. The upward phase as seen in GQ, which can be interpreted as a slow nucleotide exchange and turnover, which is known for GQ family of G proteins. A downward phase like that of the G12, which can be interpreted as a rapid nucleotide exchange and turnover, and a biphasic response as shown as the GS, which can be interpreted as a transient engagement followed by a rapid turnover. The CRC can also be drawn by taking the area under the curve of the kinetic profiles, and we can have profiles that look like upwards, downwards, and then for the biphasic, we could either take the upward phase or the downward phase, and here I've done it for both to calculate the area under the curve. And for all the data that I'm going to be showing, I have picked the one that I've thought was the best fitting, and I haven't shown you both. So for the AT1R, the GS has a biphasic response. The GI family has shown mostly a downward response with a very short up phase. The GQ family have mostly an upward phase. However, the signal from the G14 is not robust enough, and a downward phase for the G12 family as well. So AT1R is showing to be very promiscuous in its binding to the different G proteins. And similarly, I've shown all the different concentration response curves that I've plotted using the area under the curve. And then similarly, it's a very similar story. However, for GS, it's a downward phase. And for most of the GI family, we see a downward trend as well. For the GQ family, we do see a consistency with the different G proteins, the different receptors, that it's mostly an upward phase. And then for the G12, again, downward phase as well. And similarly, all the different concentration response curves. For the CXCR2, again, very similar to what we observed with the CCR2, downward phase for GS and the GI family, an upward but really transient phase for the GQ, and then a downward phase for the G12 and 13. And this was the only receptor that showed some kind of binding to G14. The others two didn't. So, and there's the concentration response curves again. And so once I optimized the assay format and system for the monomer recruitment assay, I carried out the GPCR hit assay for the AT1R with either CCR2 or CXCR2. So what the GPCR hit assay is that in this assay, we have a tagged receptor and we co-express it with an untagged receptor. When the ligand is added for the untagged receptor, any concentration response would indicate a proximity or interaction between the two different receptors and the G protein, which could indicate a formation of heteromer. However, upon carrying out this assay, I did not get any response. A parallel assay was run with only monomer format. So upon carrying out this assay, I did not get any response, and a parallel assay was run with only a monomer format to show that the assay conditions were working fine. And I tried several different optimization steps, including lowering and increasing the amount of DNA transfected and trying different ratios of each receptor, but no interpretable signal was obtained in any of the test conditions. So in summary, I optimized a breadth-based assay, which was established for probing the G protein recruitment to the target receptor. AT1R, CCR2, and CXCR2 each displayed broad G protein coupling promiscuity and recruiting 11 of the 12 tested G protein subtypes and exhibiting distinct subtype-specific kinetic profiles. GPCR hit did not detect any interpretable signal dependent G protein recruitment under the conditions that I've tested. So while my original aim was to investigate the heteromerization using the GPCR hit assay and work towards the structure of a heteromer, because of some of the problems that we faced and we needed to solve those problems, issues, and work towards it, I decided on working on determining the novel structure of the monomers of AT1R and CCR2. So which brings me to my second aim, which is to the biochemical optimization for the structural determination of AT1R with angiotensin II. So this is the general protocol of how we do our biochemistry in our lab. So we have our PFAS viruses, which we use to generate baculoviruses. Then we infect some insect cells, SF9 or high fives, to express proteins. We then use detergent to solubilize the membrane and get the proteins out of the membrane. And then we use some kind of affinity column to purify the protein. I'll be talking about these in specific details with all the preps that I'll be doing. And then after that, we do a size exclusion chromatography, after which we get our protein, and then we check it with a negative staining. And then if we have a good sample, we then go to cryo by preparing grids, and then we image it under the cryo microscope, and then we process the data. So there are some of the strategies that I will be using in order to stabilize or attempt to stabilize the receptor G protein complexes. And the first strategy is to use a dominant negative G protein. So these are G proteins with mutations that reduce nucleotide affinity and impair nucleotide exchange, which helps reduce dissociation of receptor G protein complexes. The second strategy was to use mini G proteins. These are engineered G proteins, which in which the alpha helical domain has been truncated and an additional stabilization mutation has been introduced in order for it to be more stable. I will also be using stabilizing antibodies, one of them of which is the SCFE16, which binds to the alpha N helix of the GI family of G protein, as well as the G beta gamma, and it helps stabilize the complex. And the other one is nanobody 35, which binds to the interface between the G alpha and the G beta gamma subunit of the GS family. For the AT1R work that I'll be doing, I will be constantly using dominant negative GQI and mini GSQI. So dominant negative GQI is a GQ chimera in which the alpha N helix has been replaced with that of the GI family of G protein. So this allows the binding of SCFE16 and helps stabilize the complex. And mini GSQI is a mini GS based construct in which the N-terminal region has been replaced with that of replace that of the GI, allowing it for the stabilization with SCFE16, while the C-terminal of the receptor contacting region with that of the GQ, so that its binding and functionality is that of GQ. Because the core of the construct is still GS based, it also remains compatible with using nanobody 35. So we can use both nanobody 35 as well as SCFE16 while using mini GSQI. So this was the first prep that I did. So I co-expressed the flag tagged AT1R with 8 his, including the dominant negative G alpha QI, beta, and gamma. And I was using angiotensin 2 and SCFE16. So after the after the expression, the cells were detergent solubilized, and then we used a flag column because the receptor has a flag tag, and then it goes through size exclusion chromatography, and this gives us a chromatogram which looks like the one shown here, where the x-axis represents the elution volume and the y-axis represents the absorbance at 280 nanometers. And based on the expected size of the receptor G protein complex, we can estimate the elution range where the complex should appear, and fractions from that region can be collected and analyzed further by methods like Western blot or negative staining, Cryo-EM, etc. So this is the first AT1R prep that I did, and this is what the SCC profile looked like. We collected samples from the different region of the SCC and analyzed them by negative staining. In peak 1, we could see mostly micelles and monomers. In peak 2, we saw mostly complex-like particles along with receptor-only particles. And in peak 3, we saw mostly receptor-only particles with some complex as well. For those of you who are not used to looking at these micrographs or 2D classes, here I have overlaid a GPCR complex on top of the negative stain 2D class image. However, in my peak 2, only 20% of the total particles were actually complexes. And because the yield from this prep was very low, where the complex peak was below 20 milli-AU, the sample was not suitable for Cryo-EM. So here I analyzed the different stages of the purification by Western blot to understand why the yield of the sample was so low. The 3 blots have the same sequence of sample and have been incubated in either anti-GQ, which detects the dominant negative GQI G protein, the anti-His, which detects the His tags, so the AT1R, G beta 1, and SCFE16, which all have a His tag, and then the anti-flag, which detects the AT1R. So one thing we can see from the first blot is that the dominant negative GQI is present in all the earlier stages of purification, but when we go to M1 elution, we significantly it is being significantly lost during elution. So the M1 elution lane on this His blot also shows a strong AT1R band, whereas we can see a G beta 1 band running through these 3 lanes, which means that because there's no G protein, we can also not see any elution, the G beta band in the M1 elution here as well. So we see a strong AT1R band. And this was a common bottleneck for all my dominant negative GQI purifications. And I've tried using additional GQI pellets. I've added higher SCFE16 concentrations and optimized a lot of detergent conditions, but I was still not able to get complex stable. So which brings me to my second attempt of using a different strategy, which is after multiple attempts of trying to purify the dominant negative GQI, the complex stability remained a major bottleneck. So I moved to using a mini GSQI. So just like before, I co-expressed the flag AT1R 8 his with the mini GQI, same beta and gamma, and processed through with the purification. And here I obtained an SCC profile for the mini GSQI preparation with fraction 1 to 4 collected across the main peak region. And then I analyzed it along with the M1 flag elution and SCC, all the SCC fractions by Western blot. So on the anti-flag blot, the AT1R is clearly present across the elution and SCC fractions. On the anti-GS blot, which detects the mini GSQI, we can also see the signal in the same fractions, indicating that the mini GSQI was recovered together with the receptor. And in the his and anti-his blot, the signal reflects that this his tag components are also, the G-beta one is also being detected in the blot. So importantly, from this prep, what we saw was that mini GSQI was still being able to, was still present in the SCC fractions, and in particular, the fractions which correspond to where our complex region is expected to elute. And this suggests that the complex was retained much better through SCC than what we had seen with dominant negative GQI system. However, the overall recovery was still very low. And although it was an improvement, the system was still not suitable for Cryo-EM. So once we saw that mini GSQI gave us a better retention than dominant negative GQI, we then moved to further optimize the step based on this structure paper, which was published during my PhD. In this study, the receptor was engineered with a constitutively active mutation, and it was solved using a sarlazine angiotensin 2 as a ligand. And based on this design, we generated a modified construct containing the constitutively active mutations. And also we copied the entire structural features. However, we also added a tethered mini GSQI in order to increase the likelihood of maintaining the receptor and G protein association throughout the purification, and therefore improve the recovery of a stable complex for downstream structural studies. So this construct had a flag tag, a 10S hits tag, a maltose binding protein, and a mutated AT1R with the same constitutively active mutation with a 8X his tag, and it was directly tethered to a mini GSQI. So this was the first prep that I did for the mini GSQI tethered construct. Again, same thing. We generated the baculoviruses, expressed it, solubilized it. However, this time we used a talon resin. So a talon resin is a cobalt-based affinity resin that binds to all the his tag proteins. So since both the receptor construct as well as the G protein construct have a his tag on them, that allowed us to capture both the receptor as well as the G protein. And this was followed by a size exclusion chromatography. And in the earlier attempts to purify this construct, I repeatedly observed aggregation during concentration steps and after elution. And because this construct contains an MBP tag, which has been reported in some cases to cause aggregation, despite often being used to improve solubility, I I hypothesized that this might be contributing to the problem. So there was a paper that suggested that adding maltose could decrease the aggregation issue caused by MBP. So I have added 10 millimolar of maltose in every step of this purification to see if it can help reduce aggregation. So here is the SCC chromatogram as shown on the left, with the gray shaded region indicated the expected range of the complex. I pooled the fraction from this region and then analyzed them by Comasi staining and Western blot. From the Comasi staining, we can see that there are still multiple bands present in the pooled SCC fractions, which indicate that the sample remained heterogeneous. I then performed a Western blot analysis, and the anti-MBP blot detects the MBP tagged AT1R with the mini GQ, and this is detected in this region. Similarly, the anti-GS detects the mini GSQI, and we can also detect both the mini GSQI as well as the tethered mini GSQI in this blot. And similarly, the anti-His blot detects the AT1R, G beta 1, and nanobody 35, and we can also detect all the different components from this. So upon doing the Western blot and seeing that all my components are in there, I did a negative staining on the complex peak from the SCC, and I found that around 40% of the particles were complex-like. And this was one of the best results that I've seen so far across all my AT1R purifications. So I took the sample forward for Cryo-EM, and the grids showed patchy ice and uneven particle distribution, which is a result of suboptimal vitrification condition. And although I could see some particles with complex-like shapes, the 2D classes were low contrast and poorly resolved. So at this stage, the issue was not that the complex was completely absent, but the sample and grid conditions were still not good enough to generate a high-quality particle stack for 3D reconstruction. And because I did not have enough material left to test additional vitrification condition and glow discharge conditions, and I also did not have time to generate more complexes and purify them, I could not take this workflow any further within the time frame of my PhD. So in summary, the dominant negative GQI-based AT1R workflows were bottlenecked by insufficient receptor G protein complex stability. Mini GQI improved retention and recovery of the G protein during purification, and the engineered AT1R mini GQ fusion construct further improved complex recovery, and negative stain showed improved complex-like particle purification. And sample heterogeneity and Cryo-EM vitrification condition remained a major barrier to AT1R structural determination. Then I went on with my third aim, which is the structural determination of the chemokine receptors. So here I'm going to be introducing you to some of the main biochemical strategies that has been used to determine chemokine receptor structures. The first is the chemokine added approach, where chemokine is added during purification, typically at the complex formation stage, and then it's carried out throughout the purification steps. And one of the examples is the CXCR4 structure. The second one is the chemokine co-expressed approach, where the chemokine is co-expressed together with receptor and other complex components using baculovirus express system. And some of the examples are the ACKR3 and the CXCR2 structure that has been determined using this approach. And the third is the chemokine fused approach, where the chemokine is fused directly to the N-terminus of the receptor. So both the chemokine and the receptor are expressed as part of the same construct. And one of the examples that has been employed for this is the CCR2, CCL2 structure. So one of the major bottlenecks for my project is the availability of chemokines, as they are quite expensive to buy commercially, and running multiple trials of purification of complexes is not feasible. And adding the ligand in the complex formation step would use up a lot of chemokines, and because they're very valuable, I looked at a lot of different papers and designed this new modified protocol in order to see if I can get a structure of the chemokine receptor. So what we do is we first infect our cells with all the viruses as before, and then we solubilize the complex. But before we initiate complex formation, we capture all the G protein as well as the receptor with a TALEN resin, which captures all the his-tag proteins. And then we concentrate this elute. And because now we have a very small volume, that is when we add in our ligand as well as the stabilizing antibody. And then we incubate it while shaking at 4 degrees and then perform a size exclusion chromatography. This really helps us use our ligand efficiently. And I would like to thank Martin Stone and Ram for all the ligands they have provided. So all the biochemistry I've done with ligands, they have provided us with that. So thank you. So as discussed earlier, once I got the elute from the TALEN resin, I split them into 2 different batches, and one I incubated for 2 hours before running SEC, and the other I ran overnight. So the 2-hour trace looks like it has a large complex peak, whereas the overnight incubated sample on the right might have formed more aggregates, thus the complex and the void peak are on the same level. I then did a Western blot analysis to inspect the different components of the complex. So the anti-flag detects the CCR2, the anti-GI detects the dominant negative GI2, and the HIS detects all the CCR2, beta-1, and SCFE16. And all the different components were present in the Western blots, like the receptor and all the other components. And I then took this to cryo, and the sample that I incubated for 2 hours actually gave me much better looking 2D classes. From the initial 2D classification, I was able to obtain some promising classes with visible structural features of the complex. However, I was still not able to take this data set through to 3D reconstruction. And although there were a few good 2D classes, the sample still suffered from heterogeneity and the vitrification conditions were not ideal. Sorry. I tried analyzing the dataset using different processing tools and approaches, including neural network picker, to see whether a particle picking could be improved, but that did not lead to any better outcome. So although this data set showed clear sign of progress, it was still not sufficient to generate a reliable 3D model. So at this stage, although the modified CCR2 workflow allowed me to use the chemokine much more efficiently, chemokine availability was still becoming a major bottleneck after multiple purification trials. So I moved to a different strategy where I would no longer need to keep adding chemokines during purification and could instead focus on optimizing the purification itself. To do this, I designed the next construct based on the published active CCR2 structure, which was also published during my PhD, where the CCL2 was fused to the N terminus of the receptor. So I followed the same overall design, but replaced the CCL2 with the CCL7. This construct also incorporated the nanobit tethering strategy. So in this system, large bit and high bit are 2 different fragments of the nanolock with very high affinity for each other. And so when they are fused to different parts of the receptor G protein assembly, they hold these components in close proximity to each other and prevent complex dissociation during purification. So this construct helped me address 2 issues at the same time. First, by using CCL7 directly by tethering it to the receptor, I removed the need to add chemokine during purification. And secondly, by incorporating the nanobit tethering strategy, it provided an additional means of stabilizing the receptor G protein complex together with SCFE16. So again, for this purification, I followed the same overall strategy that was used in the published structure of the CCL2 CCR2 structure. So the receptor constructs were expressed in SF9 cells, after which they were solubilized with detergent. And because this construct has a MVP tag on it, I used an amylose resin, which can pull out the MVP tagged receptors. And then once I had purified it for one stage, I then added a 3C protease cleavage. There was a 3C protease cleavage site between the large bit and the MBP to get rid of the big clunky MBP that's bound to this receptor. And then we again go through another batch of amylose resin column to capture all the free MBP, and then we get a structure of the receptor complex without the MBP tag, after which we go through the size exclusion chromatography. So after several rounds of optimization with this construct, I obtained an SCC profile that looks like the one that's shown here. The gray shaded region indicates the expected dilution range of the complex, and I pulled the fraction from this region and analyzed it with COMASI as well as Western blot. Here we can see that the multiple bands are still present in the COMASI, which shows that the sample still is heterogeneous. I then analyzed it, the same pool fraction by Western blot, and the anti-analog blot here detects the large bit tagged constructs. So we can see that we have the full length receptor construct with this MBP still attached to it, and also some free large bit that might have been separated from the receptor construct. We also have the GI2, which is detected by the anti-GI, and the beta-1, which is detected by the anti-His. And so I took this to cryo to see if I can get any structure with it, but I made a set of grids and I screened them, and the issue was again the grid quality, and the ice was super patchy, and the particles were clustered around the edges or unevenly distributed around the holes. I collected a small data set and carried out an initial 2D classification as shown on the right here, but there were some classes that looked like complexes, but many of them were still not being able to resolve. So at this stage, although the sample heterogeneity is still a challenge and vitrification has become a major bottleneck, by this point, I addressed the chemokine bottleneck and improved the purification workflow, but optimization of grid conditions has become the major limitation for obtaining a structure of the CCL7 bound CCR2 complex. And because I was running out of time for my PhD, I did not have enough sample to systematically test the different conditions, and I could not take this optimization any further. And moving forward, this would require more systematic screening of grid type, glow discharge condition, and vitrification parameters, including Vitribot settings, and to identify the best conditions that will give us a better particle distribution and support a high resolution structure determination. So in summary, the CCR2 required a modified biochemical workflow because chemokine availability was a major bottleneck for purification of receptor complexes. And optimized dominant negative GI co-expression yielded the most promising 2D classes, while with visible secondary structural features, sample heterogeneity and Cryo-EM grid vitrification remained the major barriers for structural progression. And the published tethered chemokine large bit strategy addressed the ligand bottleneck and provided a more practical direction for further CCR2 optimization, and future optimization of glow discharge and vitrification condition is required for high resolution structure determination. And with that, my future direction is to first determine whether the G protein recruitment identified in this study produces distinct downstream signaling outputs using orthogonal functional assays for non-canonical G proteins, and to further investigate the lack of interpretable signals in the GPCR hit assay by assessing the agonist induced receptor localization, quantifying membrane expression of co-expressed AT1R and CCR2 or CXCR2, and analyzing the AT1R CCR2 or CXCR2 interaction dynamics using saturation breadth, and also structural optimization to address the yield and sample heterogeneity, as well as vitrification and Cryo-EM glow discharge condition for both the AT1R as well as CCR2 to enable high resolution 3D construction. And again, to expand the structural workflow for alternative receptor G protein non-canonical complexes. For example, AT1R, GI, and G12 family are well known to couple to this receptor. And for CCR2, the GQ11 family is also known to couple to this receptor. And with that, I would like to thank everyone. Thank you, Patrick. Thank you, Denise. Thank you, Matt. Thank you, Laura. And thank you, Jason, for being super supportive during my time during my PhD. Thank you to the Stone Laboratory. Thank you, Dimerics. Thank you, Carl, for coming to my presentation today. And thanks, Robert. And thank you, everyone from my panel members and all the members of the metabolic G protein receptor biology. I don't have any personal photos, but since this is a tradition, I did have some hobbies while I was doing my PhD, and these are some of the photos I took. And with that, I would like to thank everyone. Thank you. If you have any questions.
Thank you, Alok. If you can breathe now. This is open for questions. Marcel, first.
I was curious about your the geographic activation assay in pharmacological testing. You had some traces that went up and some other that went down, and you alluded that was differences in nucleotide exchange, right?
Yeah.
So I was wondering how slow does it need to be for it to be, to become a positive? You alluded that the positive ones were being slow.
So that's one of the reasons, but so the downward response could also be caused by internalization of the receptors. It could be due to the steric hindrance caused by the beta-arrestin coming and binding to the receptors, or it could be that there was a pre-coupling, and then once we added the ligand, the conformation change would then shift the equilibrium from coupled to not coupled. So there could be plenty of reasons, but that's one of the reasons that's quite known in the literature.
Positive versus negative response is dependent on the G alpha subunit, or because it tested, I think, a few different receptors with the same success.
Yeah, definitely. The GQ family was definitely always positive. And there's plenty of evidence in the literature that GQ does have a slow turnover. I'm not sure about the timings, but it's slower. And GI generally is negative response. Yeah.
All right. Yep.
So I just have a question about the leading classes from the first approach to the CXCR2 structure.
Yeah.
So just looking at it briefly, it looks quite promising. So I was just wondering if you can comment on the number of particles you got from these two D classes, and what do you think were the reasons why you couldn't take it further to 3D classifications with this second?
So the thing about why I couldn't take it was because I think the concentration of the protein was 6.7 mg, and people generally go much, much higher. But when I made my grids, it was super packed. And after that, I did have some samples that were on the fridge that I could just thaw and reuse. But I think these samples, once you freeze them and you thaw them, it doesn't work for me. And
so, but how many particles did you end up with?
I collected around 4,500 micrographs.
But how many actual complex particles were you taking for the reconstruction?
For those were 22,000.
Yeah, so not
roughly. Yeah, 22,000. It wasn't a lot. I mean, some of them look really promising, but then when you start processing them, it's just not that useful.
Okay, we have other questions? Cindy.
A more specific question. So I noticed that you use different resins for your affinity purification that you jump from black resin to Taiwan resin. And I noticed that you have all the tags in all your receptors specified. So, and specific reasons that you choose specific affinity method in different receptor purification.
So the first reason is that when we started using especially for CCR2, when we started using FLAG, and we saw that in our elution we were losing a lot of G protein. My idea in the beginning was that if I can concentrate all the G protein as well as the receptor into the system and like kind of force them to like complex, maybe it'll be a bit helpful.
So
That was the first like.
So have you tested like systematically to compare three of those? Which one is actually better?
Definitely when you try and like pull everything down, Talon is definitely like very efficient. It pulls down all the G protein as well as the receptor, but then it also pulls down a lot of dirt. So having like a two-step purification is actually really good. And we did trial with two-step purification as well. So we first collected everything with Talon, and then we did a flag or maybe an amylose, but it's just because we work with so little protein, at the end you always get like nothing. So yeah, you do get pure protein, but
Any other questions? If not, then also substitution. As a finishing student, you get a little